Thanks a lot. It's really a pleasure to be here, to be with old friends. Quite a number of those, by the way, and to acknowledge the parcours of François, which is really outstanding. So I think I will have quite a different presentation from the other, but let's see. First of all, the title, Good Friend Doing Hard Science. Why this? Because I think on the website of INDRA you can read François Tardieu studies the connections between agronomy and hard science, analyzing the behavior of plants under environmental stress and creating models which have become references in the field. And when I look at what François did, you know, with the rigor, the protocols, and all the things he put in place, I think he's doing hard science. So it was a bit provocative from my side. Just I will have my one slide promo. It's about what we are doing today. And then I will link with what I've been doing with François. So I'm the director of the Integrated Breeding Platform. And this is really a platform to enable and support basically breeding, especially related to research for development all over the world. We have a product which is called the Breeding Management System. This is a crop information system like many already in place, but I think we are quite unique because we are a non-profit organization, and you know, we build our architecture by mixing a lot of open source codes, some proprietary modules working with VSNI, and we build a modular system that works with API. You probably heard about the BRAPI and you know the community that is outside to enable some linkage of different pieces of software one to the other. It's an intuitive system, well documented. We have about 700 users across 67 instances, and you know, we support both public and private sector. Public institution via grants, private sector through fee. So we're also quite novel in terms of the business model that we have in place. And the integrating breeding platform, we have more than 7,000 users or registered people, so it's quite a large community of breeders. And we also support a federation approach with a central instance being able to coordinate with local instances. So when you have a project with multiple institutions, national programs or partners, it's very easy to coordinate your data management. And we really believe that the use of system like this is a quality insurance for any breeding program. And I put the website here where you can find more information. But really where our interaction, or my interaction with François started was through the Generation Challenge program. And this was an initiative of the CGIAR created 20 years ago, around the 2002 and 2003. And one of those 4 initiatives, you know, was called Generation Challenge Program. And the mission was using genetic diversity and advanced plant science to improve crops for greater food security in the developing world. This was hosted by CIMMYT. It was a 10-year project, and I believe several of you in the rooms were engaged in this project, and you would agree with me this was quite a great and unique adventure. We had money by that time. It was not easy. It was more easy to get money than those days, about 15 million per year. And we're targeting drought, and this is one of the common points with François, you know, drought and physiology. They are the 2 anchor points between the two of us. And the program was really focusing on drought-prone environments, started with 18 crops to focus on 9. And I think it was quite a nice approach because it focused a lot on a mix of competitive and commissioned projects and a mix of partners. I think the partnership was one of the highlights of the project or the initiative, including CG Centers, National Program, Advanced Research Institute, and universities. And from France, you know, we had a consortium between INDRA, CIRAD, IRD, and was one of the funders of the Generation Challenge Program. And we were really a broker in plant science, bridging the gaps between upstream and applied science. And you have here also a couple of websites where you can find some legacy history of the program. So the first interaction with François, a couple of examples, and I will talk about here through the Generation Challenge Program. Yesterday night I had dinner with François, and we were thinking what happened before the Generation Challenge Program. And luckily we had some glass of wines, and the more glass of wine we had, the better we were to identify where the story started. And it started, you know, with a meeting in Serbia with Steve Quarry around the 1990. And this is when I met François for the first time. So the belief that alcohols make you lose your memory is completely wrong. I mean, because with more glass of wine, more accurate we were. by the way, we need some rep. I mean, you know, one rep is not good enough, so let's repeat the experiment as a good scientist. François also supported my application when I applied for director. And, you know, I find those 2 emails. This is the oldest one I have from François. And the first one is Jenny, the secretary of the GCP, acknowledging receiving the letter of support from François. And below, not sure if this is, I don't take out the risk. Is a pointer? Yeah. Pointer, this is a message from François. The letter is arrived, good luck, F. So I thought, gosh, I hope he had a bit more meat around the bone for the support letter. And apparently he did. He did a good job because I got the job. So, but it was first interaction. And again, here you just have, you know, some of the novelty and uniqueness of the Generation Challenge program by the time. The second one is that, you know, I really also learned that François was quite a bold guy. This is a message he wrote to me in June 2005. And of course, I will read in French. Je t'écris à propos du papier que nous devons écrire pour Journal of Experimental Botanic à la suite d'Internaute. Personnellement, j'en ai ma claque d'utiliser les papiers de synthèse pour expliquer que c'est bien d'utiliser la modélisation pour analyser les variables génétiques. 3 papiers dans ton bouquin, ça suffit comme si. Voilà. Alors d'abord, vous remarquerez, you will notice that, you know, François tout petit déjà était très coquin. Des petits jeux de mots comme ça à la fin de ses messages. But the most important that he was directly going to the point and his idea was quite clear and transparent. More from a scientific point of view, I mean the scientific collaboration with François, he actively engaged with a number of projects in the Generation Child program. He was the principal investigator of 1 competitive grant. I will come back on this, and was engaged in several training courses, and he was also engaged in the list of other projects. I will not go through them individually, but you know, you can go through it. And quite a number of people in this room, you know, were also engaged in the different research activities that were conducted by that time. And I said, you know, that the scientific link with François was, again, physiology, drought, and I wrote, in fact, before I became the director of the Generation Challenge program, and applied as a scientist at CIMMYT for several competitive grants, and I got this one, and this was originally in 2003. I was the PI of this one. Then I became the director of the Generation Challenge program. So basically, I passed this to Mark Sokin, and then François kindly agreed to take on the leadership of this. And it was an interesting project that was related to candidate gene. It was looking about, you know, comparative genomics, which was one of the pillars of the JCP, across different plants, maize, rice, and wheat. Of course, it was related to tissue growth, and it was also related to leaf expansion across 3 organs: leaf, silk, and root. And this was focusing on maize. So the QTL detection was quite the basis. I mean, I was an ASI guy. He was a leaf grow guy. So we said, why don't we just look at this and see if there is some common basis? This was basically the rationale of this collaboration. And of course, you know, he also worked with ERI by the time to characterize a bit what was going on in rice. And the outcome, I think, were quite interesting. So it is definitely to see that there is some potential genetic basis for those different tissue, the growth of those different tissue that we looked at. And also the fact that, you know, it is interesting to compare the different approaches under field and control condition. I was taking, by the time of the field, he was taking care of the controlled condition. And the conclusion of this project, you know, came out in a paper where Claude, who also, of course, was actively engaged in our collaboration and research, wrote for Journal of Experimental Botany. This is a famous paper in the message of François before. We need to write this paper in Journal of Experimental Botany as part of a consequence of interdrought. And I think, again, that is quite a discovery by that time, you know, that there is a potential and a good possibility so that you have common genetic bases for some of those organs. And as I said, the last sentence is quite relevant. If this result is confirmed by further study, it may have profound consequences both for the modeling of the genotype by environmental interaction and for designing drought-tolerant isotope. And I think this was the first kind of, this was in 20 years ago, the first kind of thought about how can we dissect those, what we believe were these drought silo, these drought regions on the genome where you had a number of genes involved in the growth or the regulation of different drought mechanisms that were linked, you know, and how do you break this linkage drag? And I think this was an important thought, and we tried with Alain Charcosset and the meta-analysis and Biomercator to try to dissect a bit some of those traits related again to silk, ear, leaf, and some other water potential and other character, and try again to be able to cut this genetic link and be able to make progress and be able also to predict what a certain genotype and how it will perform under specific condition. François also came and with his background as a physiologist also insist on the fact that you must have good phenotypic characterization if you want to do genetics. I mean, genetics, by that time, a lot of people were excited by association genetic, the prediction, gene discovery, and all of this. And the message from François was very clear: phenotype, 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 and quality phenotype. And therefore, he contributed to several courses. One of them he organized, I think, was here at the beginning of the Generation Challenge program. Of course, it was, as expected, a mixture of lecture, practical work, and computer session. You already put quite early, you know, this idea that you need also to put your data, to model, to play with the data, to see exactly, understand what it means. And again, this has been really a good contribution. And again, we insisted during the entire life of the GCP that good and reliable phenotype was the basis for whatever else you want to do in crop improvement. This is just also a contribution of François to one of the books that I put together in 2000. I think, you know, you think that in your career you need at least once to make a book, right? And if you do more than once, that your mental health should be questioned. And if at the end you are declared healthy, mentally healthy, then you're definitely a strong masochist. So I did one only, and you know, I think the relevance of this was that it was really a combination based on the review conducted by Pat Byrne, that it grouped quite a number of different fields and economic physiology, statistical modeling, genomics, etc. And again, I think Fred Van Owyk and Graham and others were already contributing to this book. And François had a chapter, of course, surprise, surprise, leaf growth under water limited condition. And I think quite some interesting message came out of this. You know, first, I mean, he really warned, and that was a bit of evidence, but I think it was good to put again into context that to identify the respective role of each gene involved in primary mechanism or signaling pathway will take time. So that's, you know, you need to be patient and to accumulate quite a number of data. The other interesting, I think, finding or conclusion or report in this book was in this chapter is that most likely the main advantage when talking, let me start from the beginning. Although identification of QTL of parameters of response curve to environmental condition is more time consuming than analysis of raw phenotypic trait such as leaf length, leaf area, or yield, it has several advantages. And most likely the main one is that this method characterizes QTL of the behavior of genotype per se and not the behavior of the genotype in a given environment. And I think that's quite true. And of course, it was promoting his business that modeling is also a nice complementary way to move forward. A proper phenotypic analysis should also result in a phenotypic footprinting, namely in a vector of parameters of ecophysiology model that characterize each genotype. And again, going to this notion of prediction. How can you predict the behavior of a specific genotype? I will arrive to my last slide. First, you know, the characteristic of François. A builder, without any doubt, he's a team building. A team builder, I mean, he works as a team with partners and colleagues around him. He's a very didactic teacher, and I think that has been very useful on several occasions for the GCP, a rigorous scientist, and of course with a world reputation. And I just mentioned the prize he won with his team in 2014. He's an innovator, really. His ancestors were definitely working with those guys, having innovative way of growing maize. Pioneer in ecophysiology, visualization of plant modeling. I don't know if you talk to a modeler, I understand normally 50% of what they said, but with François I was able to reach the 80%. So I think he was quite good in vulgarization. And science and the service. I think the M3P has been one good example of how you can take advantage of the service, working with experts without having to repeat or build your own lab and repeating the wheel, as it's too many times happened in the public sector. And he was a coordinator of Phenom. But perhaps the most outstanding one is, again, François as an outstanding person. I mean, when you can go on the website currently of INRA, you can read that François has 61.5 children. So definitely he has outstanding capacity in applied genetics. And he has special powers because he's able to do half kids. So this is quite unique. So I'm going to write to the direction of INRA A and to change the picture of François, and definitely this is the right one to put on the website. And this is the last one. This is a picture of the Generation Challenge Program annual meeting in 2006 in Brazil. And here is François in the middle of the group. Thank you very much.